So this is the question paper, and this video, the review of the answer keys of this question paper will be there. The first question is Koch's postulates. Second is enlist three anaerobic culture methods. Third question, write three methods of demonstration of bacterial capsule. Fourth, define pandemic. Give examples of pandemic occurred in India. Fifth question is mention six dimorphic fungi. Sixth question, list out antigen detection methods for diagnosis of parasitic diseases. Question number 7, plasmids. Question number 8, write examples of intrinsic antimicrobial resistance. Ninth question, enlist any three modes of transmission of bacterial infections with examples. Question number 10 is inclusion bodies of viruses. So we'll see the answer keys of these questions. So this was the exam conducted on 29th for MBBS candidates. So Koch's postulates. So what was the necessity of Koch's postulates? To prove that particular organism is the causative agent of that particular disease, because in those times where a large majority of the reports were coming to claim that particular organism to be causative agent of that disease. So Robert Koch, he has postulated that number one in this is organism should be constantly associated with the lesion of the disease. Number two is it should be possible to isolate the organism in pure culture form. Number three is after inoculation in the appropriate laboratory animals, it should produce same disease or similar lesion. And fourth is isolation of the same organism from the lesion in the animal. Then after subsequent development of immunological methods, fifth criteria was added, it should be possible to demonstrate the antibodies against the causative agent of the disease. When the molecular biological techniques develop, molecular biological criteria is added, that is, it should be possible to demonstrate the genome of the organism. And there are so many exceptions for Koch's postulates. For example, for gonococcus, there is no any animal model. It's not possible to cultivate gonococcus in any animal. And Treponema pallidum can be cultivated. So this is uncultivated. Mycobacterium leprae is another exception where it can't be cultivated. So Treponema pallidum, Mycobacterium leprae, they can't be cultivated. But although they are the causative agent of the disease, respective disease. And gonococcus, like Neisseria gonorrhoeae, there is no animal model. Then anaerobic culture methods. Macintosh fields anaerobic jar, Anaxomat, gas pack system, anaerobic glow box, use of reducing agents such as glucose, thioglycolate, and Robertson's cooked meat medium. So these are the anaerobic culture methods. Next question is methods of demonstration of bacterial capsule. Negative staining, McFadden capsule stain. McFadden capsule stain is used for demonstration of capsule of Bacillus anthracis. Quellen reaction, it is also called as capsular scaling reaction, where antibodies against the capsular material is added, kept it for 30 minutes. After that, this wet mount is prepared. The capsules will become big refractile. So that is a Quellen reaction. The detection of capsular antigen by latex agglutination test. So these are the methods of demonstration of bacterial capsule. Pandemic. Next question. Infection that spread rapidly over large area of the world is known as pandemic. So all of you know COVID-19 in year 2020, more than 200 countries were involved. Then cholera, a seventh pandemic is occurred so far. Influenza, swine flu in 2009, H1N1 was there. Then plague, three pandemics happened so far. Pandemic was last. Then dimorphic fungi, enlist the dimorphic fungi. Histoplasma capsulatum, Blastomyces dermatitis, Coccidioides simitis, Paracoccidioides brasiliensis, Penicillium marnefi, Sporothrix schenki. So these are the six dimorphic fungi. All of you know dimorphic fungi. The fungi shows two different morphological pattern. One is as 37 degrees Celsius, they are in yeast forms, and at room temperature or in the culture media, they are in the mold forms. So these are the causative agents of systemic mycosis. Then now antigen detection methods of for the diagnosis of parasitic diseases. This is little bit challenging to remember because over a period of time the antigen detection methods are developed. Previously only direct microscopic examination was followed. So now for example amoebiasis ELISA, in which 170 kDa of lectin antigen in blood and stool, 170 kDa of lectin antigen in blood and stool is detected. Then immunochromatography for Giardia, alpha 1 Giardin antigen. Alpha 1 Giardin antigen is being detected. Entavasculetica entavadispar 29 kDa antigen. Then cryptosporidium isomerase antigen is used. So please remember immunochromatography, Giardia, alpha 1 Giardin antigen. Entavasculetica 29 kDa antigen and cryptosporidium isomerase antigen is used in the format of immunochromatography. Then malarial parasite histidine rich protein 2. So especially it is used for diagnosis of Plasmodium falciparum. So what is the important thing in Plasmodium falciparum? This HRP2, it can be used for diagnosis, but it can't be used for prognosis. All species of malarial parasites, PLDH, that is parasitic lactate dehydrogenase, and aldolase based immunochromatographic kits are available, where we can detect all parasites of the all malarial parasites, other species also. Especially if you are suspecting Plasmodium falciparum, you have to go for HRP2. Then lymphatic filariasis, ELISA immunochromatography test also are available for filarial antigen using monoclonal antibody against OG4C3 and AD12. Please remember this OG4C3 and AD12. OG4C3 and AD12 antigens. Now come to the plasmids, extra chromosomal double-stranded DNA. It's not essential for survival, or it may be single or many independent replication. Episome, it may get attached to the genome of the bacteria, or it may get detached from the genome of the bacteria. Then there is a procedure known as curing, getting rid of it. If you treat with acridine or thymine, the plasmids are removed from the bacteria. So that is called as curing. Then classification of plasmids: conjugative plasmids, non-conjugative plasmids, compatible or incompatible. Then what is the function of plasmid? Fertility plasmid. So it will produce the special type of embryo with which it joins with the F negative bacteria and transfer. There is formation of hollow conjugation tubes. So that fertility plasmid that encodes for the development of special embryo. Then R plasmids, the plasmids which are encoding for the drug resistance genes. Then col plasmids, the plasmids which are encoding for the colicin production. Then virulence plasmids, E. coli, sidrophore, adherence antigens. So these are the plasmids which are encoding for certain virulence factors for the bacteria, which will give the survival advantage to bacteria. Then metabolic plasmids, which will modify the metabolism of the bacteria. Then plasmid is also used as a vectors in genetic engineering. So this is little bit short in about the plasmids. Now come to the intrinsic antimicrobial resistance. So you can see intrinsic resistance means it is the resistance by because of the intrinsic metabolic pathways of the organism. So there is nothing like it's not acquired in the future. So it's intrinsically present. So you can see Enterobacteriaceae, members of family Enterobacteriaceae are intrinsically resistant to the antimicrobial specific for gram-positive organisms such as clindamycin, daptomycin, fusidic acid, glycopeptides like vancomycin, lipoglycopeptides, otravancin, teicoplanin, and telavancin, linezolid, tedizolid, quinpristin, dalfopristin, rifampin, and macrolides, erythromycin, clarithromycin, azithromycin. So this Enterobacteriaceae family, they are intrinsic resistance to these antibiotics. But there is little bit exception. Salmonella and Shigella, they are susceptible to azithromycin. Then come to the Klebsiella pneumoniae, same as Enterobacteriaceae plus ampicillin and ticarcillin. So Klebsiella pneumoniae along with above these antibiotics, in addition to that ampicillin and ticarcillin is there. Then Citrobacter species, same as Enterobacteriaceae plus ampicillin, first and second generation cephalosporins, cephomycins, amoxicillin clavulanic acid, and ampicillin sulbactam. Enterobacter species, same as Enterobacteriaceae plus ampicillin, first generation cephalosporins, cephomycins, amoxicillin clavulanic acid, ampicillin sulbactam. So these are the intrinsic. Next again, so many are there. Then non-fermentative gram-negative bacteria. Non-fermentative gram-negative bacteria are intrinsically resistant to penicillin. That is benzyl penicillin, cephalosporins, cephalosporin 2, cefuroxime, then cefamycin, cefoxitin, cefotitan, clindamycin, daptomycin, fusidic acid, glycopeptides, linezolid, macrolides, quinpristin, dalfopristin, and rifampicin. Pseudomonas aeruginosa, same as non-fermented gram-negative bacilli plus ampicillin, ceftriaxone, amoxicillin, clavulanic acid, ampicillin, sulbactam, ertapenem, tetracycline, stagicycline, cotrimazole, and chloramphenicol. Acinetobacter baumannii, same as NFGNB plus ampicillin, amoxicillin, clavulanate, ertapenem, astronam, chloramphenicol, and fosfomycin. Strophomonas multifoliae, same as NFGNB plus ampicillin, amoxicillin, cefotaxime, ceftriaxone, cefepime, amoxiclav, astronam, imipenem, meropenem, ertapenem, polymyxin B, and colistin, aminoglycosides, chloramphenicol, and fosfomycin. Then gram-positive bacteria. Gram-positive bacteria intrinsically resistant to astronam, polymyxin B, colistin, and nalidixic acid. Staphylococcus aureus, same for the gram-positive bacteria. Enterococcus species, same for the gram-positive bacteria, plus cephalosporins, aminoglycosides, clindamycin, and cotrimoxazole. So these are all intrinsic resistant. Now come to the ninth question: modes of transmission of bacterial infections with examples. So this is a simple question. Multi-drug resistant organisms such as Staphylococcus aureus, E. coli, Klebsiella transmitted by the direct contact. So here the hand washing precautions, hand hygiene, maintenance of hand hygiene is very important. Then droplet, meningococcus, corybacterium diphtheria, pneumococcus. So droplet. Aerosol transmission, mycobacterium tuberculosis. Ingestion, salmonella, shigella, vibrio, diarrhea, E. coli. Vector-borne, rickettsia, borrelia, plague. Sexual contact, gonococcus, treponema pallidum. Vertical transmission, mother to baby, treponema pallidum. Birth canal, listeria, streptococcus agalacti. Then come to the last question: inclusion bodies of the viruses. Aggregates of the virions useful for the diagnosis. Location, intracytoplasmic, for example, pox viruses, rabies viruses. The name of inclusion body for rabies virus, that is called as Negri bodies. Then intranuclear, in 1934, Quadri was the name person who classified into Quadri type A, which is having variable size and have granular appearance, and Quadri type B, more circumscribed, amorphous, and hyaline spheres, multiple in numbers. So these two, Quadri type A and